Recording's already?
Yes.
Yeah, great. All right, so sounds like we are ready to get started. So welcome everybody. This inaugural seminar of the Digital Futures Femtech and Feministic Focus Period. The focus period is going to run over the coming 5 weeks. Here at KTH, it's organized by researchers from Stockholm and KTH, and it's going to bring together an amazing set of people from around the world, and also locally, as today. So I'm Aira Lambert from SU, and it is my great pleasure to introduce our first speaker of the focus period, Schuppe Koistinen from Karolinska. So we get to bring together the Stockholm trio here for some really fascinating conversations. The presentation is being recorded, but we're going to turn off the recording then for the conversation after the seminar. So have your questions ready and be prepared for a really interesting talk and a really interesting conversation to follow from that. And with that, over to you, Ina. Welcome, and thank you for joining us.
Yeah, thank you very much for your introduction, and I'm really happy to be here, and I think that's an amazing initiative you have started here. And so I'm going to share some insights into the research we are doing at the microbiology, tumor and cell biology department at the Karolinska Institutet. So I will be talking about the microbiome and what role the microbiome plays for our health, especially women's health. And I was not very sure about how much you know about this field. So I will start very brief to introduce you into the world of the microbiome and talk a little bit about the gut, and then I will focus more on the vaginal microbiome and what we know about its role in women's health, fertility, and pregnancy. So we are actually born 100% human or sterile, and during the birth process we get colonized with bacteria and microbes. And just to introduce you into this kind of area, the way we measure is that we do sequencing. So that's my kind of illustration for this tree of life with the DNA strands and the world of. And the microbiome is the microbiota are all those small microbes that live in our bodies. So it is both bacteria, but also viruses, archaea, fungi, and other one-cell organisms we have in our bodies. The gastrointestinal tract is actually one of the most densely populated microbial ecosystems in the world. We have about 1.5 kilograms of bacteria in our gut. This is almost like its own organ and the same weight as our brain. And these bacteria encode, or microbes encode, an enormous number and outnumbers our human cells. And we have most of them live in our friends. So we have never been on Earth without living in symbiosis with microbes. So they support our health, and I will explain a little bit more about that. So, and our research focuses on how that microbiome affects women's health and reproduction. So when we are born, we are still sterile in the womb. And it has been a very large controversy in the academic world if we really are sterile or if the placenta harbors a microbiome. But during the last couple of years, we could agree, and we also contributed to that with a paper that the fetus is sterile, but gets colonized with what we call vertical transmission during birth. And then, of course, it makes a difference if you are born through the birth channel and get in contact with the vaginal microbiome, or if you are born with C-section and get in contact first with skin microbes and the microbes from the hospital environment. But there are many sources of microbes. For instance, breast milk contains like 200-300 different species of beneficial microbes that are actually selected from our immune system and transported to the breasts, and the baby gets fed with those beneficial microbes already from the beginning. So during the first 2 to 3 years of life, each individual establishes a microbiome that then during the course of life is relatively stable. And a healthy gut microbiome is also if you get infections or get treated with antibiotics or drugs, it of course has a very big variation, but it then has this resilience. So it kind of jumps back to its original state. And if it doesn't do that, then we talk about dysbiosis, and dysbiosis is connected with a number of disease states. I will be coming back to it. There are many factors that influence the microbiome. We know some of them, the others we don't. But the most important is still antibiotics, because they wipe out all kinds of microbes and bacteria, and not only the ones they are aimed for. But also our modern lifestyle with giving birth with C-sections, don't breastfeed, a diet that may not be the one we are developed to live with, hygiene habits, less contact with nature, etc., etc. But also the environment we live in, living in smaller families than before, having less contact with animals, but more contact with chemicals, for instance. And there's also a component of genetic inheritance and an association to different diseases. So the most studied microbiome is the one in the gut, and here a high diversity is connected to good health. So the more different types of species you have in your gut, the more it is considered to be a healthy microbiome. The thing that is so amazing is that you actually have a possibility to influence it. I mean, your genes you are born with, and you have the risk factors you are born with. But when it comes to your microbes, you can influence them by their lifestyle choices, mainly diet choices. And we also are very, very individual. In genes, we are like 99.8% similar to everybody. But when it comes to the microbiome, it's only about 30%. So we have very individual pattern. And at this point, we don't really understand what is the composition of a healthy microbiome, because it is so varied. And but it kind of depends on the gene functions that are present. But we don't really know all of that yet. And another important role of the gut microbiome is that it's a reservoir for a large variety of small molecules that are taken up into the bloodstream and transported to the different organs in the body. And we talk about this gut-brain axis, gut-skin axis, gut-lung axis. So the microbiome is like our inner pharmacy that influences our health and also communicates with all different parts of the body. And we are just starting to understand these connections. So the main function of all the microbiomes we have on our bodies, we have microbes on all our kind of surfaces and on the skin, in the mouth, in the gut, in the vagina, and like every surface, we have microbes, and their main function is actually colonization resistance and to prevent infections. So if you have like a very stable ecosystem, it's very difficult for other microbes or pathogens to establish a colonization. And for that, they produce different compounds, antimicrobial compounds. It can be acids like lactate, or it can be small peptides like bacterioceins. They also have a very close connection to our immune system, and they are important to keep the barrier function of the gut. So when the barrier function is compromised, we talk about this leaky gut and then low-grade inflammation that is connected with many of our lifestyle diseases. The gut microbiome helps to digest food, produces certain vitamins, which we can't produce ourselves or get through the diet, and also produces metabolites like short-chain fatty acids that are very important to keep this barrier function. The secondary metabolism of bile acids is by our gut microbiome, but they also contribute to metabolize drugs and other xenobiotics and affect different organs. I didn't say that, but I'm actually also an artist, and all these kind of colorful illustrations are watercolors. So there is one hypothesis which is called the hygiene hypothesis. And here is this observation that with the start of using antibiotics, all these infectious diseases have more or less disappeared, or they are treatable. But at the same time, in the same time span, autoimmune diseases are actually exploding. And it is very kind of prominent in the Western world, connected to Western lifestyle and the use of antibiotics. And one of the pioneers in the field is Martin Glaser from New York, and he has studied very much in detail how antibiotics affect the gut microbiota. And this is like more a hypothetical theme, but that explains very well that the kind of modern societies, on like the US in blue, that by the intake of antibiotics, we kind of lose certain taxa that used to be in our gut. And if you compare that to populations in the rainforest or other kind of less developed areas in the world, the variation of the gut microbiome is much, much larger. So that's what is called the hygiene hypothesis. And Martin also wrote a book about that, which I can recommend, which is called Missing Microbes. And I have illustrated more that we are going from like this tropical garden with a really, really rich ecosystem with both animals and plants, and that we kind of, by using too much antibiotics, by having this high hygiene and the diet that doesn't kind of support our gut microbiome, we kind of convert this tropical garden into like a savanna or a really poor ecosystem. And yeah, and when we come to the vaginal microbiome, then we actually think very differently. So for the vagina, less diversity is more healthy, as long as it is colonization with lactobacilli. So lactobacilli are the superheroes of the vagina. They help us to keep the pH low and also produce like lactic acid. And that again prevents from infections and from pathogen overgrowth. And they also kind of compete for nutrients. They adhere very, very tightly to the vaginal membranes. And they kind of have a very tight interplay with the local immune system. So lactobacilli are very important for our health. Another thing to consider is that humans are distinct. There are actually no other species that have a lactobacilli-dominated vaginal microbiome, not even human primates. And that makes it kind of difficult to study because there are no animal models that easily could be used for that. And so why is it important? Yeah, I mean, if we have a vaginal environment that is deficient in vaginal lactobacilli, we just have a higher risk for diseases and adverse obstetric outcomes. So it's a higher infection risk for both STIs, but also sexual transmitted infections, but also viral infections like HPV and HIV. For HIV, the risk increases with about 60% getting infected. And if you have a dysbiotic vaginal microbiome compared to a lactobacillus dominated, also a higher risk for infertility, pregnancy complications such as miscarriage and preterm delivery, and also pelvic inflammatory disease, malignancies related to HPV, like the development of cervical cancer and urinary tract infections. And when I talk about dysbiosis, I refer to the overgrowth of non-lactobacilli species, and they have funny names like Gardnerella vaginalis, Pravotella, Fanihessa, Mobilumpus, and others. And it can be both a mix of anaerobic and aerobic bacteria. And one of the characteristics is also that they produce this polymicrobial biofilm, so they are really, really difficult to treat. So briefly on the research we are doing in our center. So we kind of ask ourselves very fundamental questions in the beginning, because when we started that work about 10 years ago, there was very little known about the vaginal microbiome. So we looked at what is the effect of the menstrual cycle and what effect do hormonal contraceptives have on the microbiome. And these shortenings here are the names of studies, and I will be coming back to those. What composition of the vaginal microbiome is typical in healthy women, especially in a Swedish or Scandinavian population? There was very little data available. What role does the vaginal microbiota play in HPV infection and in the development of cervical cancer? And then is there a profile that associates to pregnancy complications? And we focus on prediction of preterm birth. So to talk about the first study, our ME-MENS, or the microbiome and menstrual cycle and contraception. This study is performed in collaboration with Henriette Sverre Nielsen at the University of Copenhagen. So we had 150 women that were enrolled, 50 of them using an IUS with hormones or progesterone, and then 50 women using combined oral contraception, and then 15 not using hormonal contraception. And we invited those women at 3 different occasions, at day 1 to 3 of menstruation, during the follicular phase, and then later during the luteal phase. We collected saliva, feces, rectal samples, an endometrial biopsy, and vaginal samples, and also other samples. And most of them were collected at these 3 phases, but the vaginal samples were collected during this month on a daily basis. And we connected that with questionnaires and a diary. And so just to get an overview of the data, here we have a plot of all the different types of microbiomes. So the saliva samples in orange. Then we have the fecal samples. We have the vaginal samples. Here you see they are much, much more spread than the other ones. And this part is the kind of lactobacillus dominant, and then the more dysbiotic samples, and the rectal samples that are a little bit in between the rectal or the vaginal and the gut samples. And the finding from this first paper is that the menstrual cycle phase, but not hormonal contraceptives per se, are associated with the vaginal and oral, but not the gut microbiome composition. So the gut was not affected at all during this time of mens and from the oral or the hormonal contraceptives, but we see some changes in the vagina and the oral cavity. So when it comes to the oral microbiota, we looked into that separately and published a paper here. And as I already said, the menstrual cycle phase affected. And we have some species that are more pronounced during the menstrual cycle phase, in the luteal phase. And these are connected to a more inflammatory state of the oral cavity. And I think most women actually recognize that just prior to menses, we have a very kind of distinct feeling and a much more sensitivity in the mouth. And so we could measure those species, and we saw a kind of significant difference in abundance. And when it comes to the vaginal microbiota, and here we looked over the time course. So this is the first time point, menstrual phase, follicular phase, and luteal phase. And then we look at the different species and the abundance, how they change over time. And we see the good guy in the vagina is Lactobacillus crispatus, but also other Lactobacillus species. So at menstruation, there is a very big variation, but the vaginal microbiome in most cases stabilizes, and there is higher dominance of Lactobacillus species later during the menstrual phase, while then the abundance of these pathogens kind of decreases. And that is kind of makes sense, because when there is blood in the vagina, it also increases the pH, because that has a neutral pH. And it also adds a lot of nutrients that are different from the glycogen, that is kind of the preferred nutrient for lactobacillus. And we studied this then in much more detail. And when we looked at individual subjects, and here we have representative trajectories of vaginal microbiomes from different individuals. So the blue ones are Lactobacillus, the dark blue is Lactobacillus crispatus. And here is one individual, and we have samples over 30 days. We also have information about menstrual bleeding and sexual intercourse. And then we see there are very different patterns. Some women are super stable, independent on menses and sex. And other women, they have this kind of dysbiosis. This is Gardnerella vaginalis, that comes with menses and otherwise a stable microbiome. There are women that are super dysbiotic all the time, even if the composition can change. So, and in the scatter plots, we clearly see. So this is the Crispatus one or Lactobacillus staying there. Others move in and out, and there are some that kind of are in the dysbiotic state all the time. And we kind of developed a new characterization system, which we call the vaginal community dynamics. So it's like a tool that categorizes, but you need kind of longitudinal data. And we made the kind of conclusion that we need at least 4 data points during a menstrual cycle to being able to categorize these women, if they are constant, eubiotic, or if they have the different categories I already talked about. And so this is a classification tool that is also available to everybody. And why is it important? I mean, here is just a different visualization of the composition of the vaginal microbiome, which is called the corona plot. And this is the same individual, the same woman, and that is very close to menstruation, and that is 4 days later. And here you see there are all kinds of bacterial species we can measure in the vagina. And then 4 days after menstruation, it's dominated by Lactobacillus iners and Lactobacillus crispatus. So it is important when you do a clinical study or if you measure in the vagina that you keep track of menstruation and that you are aware of that variation. And that was not really known before. And another study, we have set up a cohort in collaboration with Mats Olofsson at Akademiska Hospital in Uppsala, where we kind of piggybacked on cell screening. So we collected from women that were called to cell screening, and we asked them to donate a sample at some specific clinics, and also have information from questionnaires they answered, and also registry information on drug prescription, on childbirth, and HPV status, etc. So we started with a very big population, and in the end, we had 1,554 women, where we have both vaginal samples and questionnaire data. And just to overview the data here, because as I said, we didn't have much information about the Swedish population, that about 64% of the women are dominated by Lactobacillus. And for us, the cutoff for dominance was 60%. Here in green, Crispatus, iners, and then the more like dysbiotic bacteria like Gardnerella vaginalis in this purple color. And but only 11% of the women actually reported vaginal symptoms. And vaginal symptoms would be like itching, smell, so the symptoms of bacterial vaginosis, but also symptoms of yeast infections, for instance. So we have a very clear proportion of dysbiosis, but only 11% report symptoms. And the diagnosis of bacterial vaginosis is really a clinical diagnosis. So the question is, these women that don't have lactobacillus and dominated vaginal microbiome, but don't report symptoms, are they at risk to have lower fertility and pregnancy complications? And at this point, we actually don't really know. And there's also a big difference between different ethnic groups, and Black American women have more dysbiosis than Caucasian women, also differences in African populations and Asian populations. And at this point, nobody really knows. But it really needs to be studied in the context of risk and outcome, and not only of kind of mapping what is there. And one of the kind of take-home messages from this study is that the vaginal microbiome actually changes with age. We have a population from 23 years to up to 70 years. And the green ones here are the lactobacilli. So they really decrease. And then at the time of menopause, which in Sweden is around 51, 52 years of age, we see a really rapid decrease in the beneficial lactobacilli in the vagina. And when it comes to childbirth or have a pregnancy and a child previously, we also see that actually the first pregnancy leaves a pattern in your vaginal microbiota. So the level of, or the abundance of lactobacillus species really decreases after the first child. And there is no really good explanation for that yet. So Back to our list of questions, and the next question here is, does the vaginal microbiota play a role for the development of cervical cancer? And the first study we did was with a subset of this vermilion, where we looked at HPV expression in the samples and then compared HPV positive versus HPV negative women. And we did both typing for HPV and sequencing, and the samples showed that there is a higher diversity and higher dysbiosis in the HPV group. But we actually don't really know if it's the hen or egg here, but we know that this is a risk for development of cervical cancer. And there are certain species that are in higher abundance in those women. We also repeated that with a bigger cohort and with a different sequencing method, which I didn't talk about in detail. Shotgun sequencing versus 16S amplicon sequencing. So we have the same finding in a kind of bigger cohort, that there are some of these pathogenic species that are in higher abundance in the women that have HPV, and also in women that have dysplasia, which is kind of the first step into the development of cervical cancer. So this is a very important, we published that paper last year. And then I would like to talk a little bit about pregnancy risk and preterm birth. So we started that work with a systematic review, one of our students, where we looked at 17 studies that were published between 2014 to 21, and reanalyzed the microbiome data, vaginal microbiome data, and the conclusion of this study was that the microbiome is predictive of preterm birth, and lower abundance of lactobacilli are associated with higher risk, and women with higher or with Lactobacillus crispatus dominans are at lower risk. And later there were two more reviews, systematic reviews, that confirmed those findings. And we set up a study that we called the Swedish Maternal Microbiome Project. And here you see our colleagues and collaborators in that. And the aim of this study was to assess the association between the maternal microbiome during pregnancy and postpartum, and the risk for both maternal and neonatal outcomes. And first research question is on, are we able to predict preterm birth in this? And the study is, the data sample collection is finished, and we collected data prior to week 20 of pregnancy, so in early pregnancy, from the mouth, the vagina, and the gut. And then we also collected between week 28 and 30, and then 5 weeks postpartum. And women also filled in a very comprehensive questionnaire at each time, and we also have the possibility to look at registered data. And so this is, it was a really, really big effort from our center, and we did all the recruitment via social media. And we were really, really happy that we could reach all parts of Sweden. And the sampling was home sampling, so we sent out the kits, we got them back. And in total, we recruited 5,400 about women, and 3,100 completed, so where we have all the free time points and samples collected. We compared that to the Swedish pregnant population registry-wise, and the cohort is relatively representative, but we have women with higher education, normal BMI, and that are kind of a little bit more in our cohort than in the general Swedish pregnant population. And so if you're interested in the cohort description, this is published, so you can look at that. And I will just briefly say what possibilities we have with that study. So we have about 200 preterm birth cases, which is relatively rare in Sweden. So that's why we needed to have this huge study. We have early birth cases. We have looked at the possibility to look at miscarriages, preeclampsia, gestational diabetes, high blood pressure. So we have in total about 700 women that have complications. and also pre-existing diagnoses like polycystic ovary syndromes, endometriosis, recurrent pregnancy loss, etc. So this is kind of a gold mine to study in the future, and we are just starting with that. And I just want to show you some of the data regarding the prediction of preterm birth. And here we kind of we sequence both the vaginal and rectal samples, and we also included our metadata in it, so individual risk factors reported by those women. And when we did different types of microbiome analyses and then machine learning approaches to predict preterm birth. And this is just a summary picture. So if we only base the prediction, and this is random forest based, so if we just look at the vaginal microbiome, it doesn't really predict very well. The fetal microbiome in isolation doesn't predict very well either. The metadata or the questionnaire data of the women has a little bit higher prediction. And then if we combine all the taxa and the metadata, the prediction gets even better. But if we then actually add into this denoised model, we add alpha diversity. So how does the microbiome composition change between time point 1 and time point 2? Then we get a very nice prediction value. And we interpret that women that develop or that deliver preterm, that their microbiome doesn't adapt to the pregnancy in the same way as in the women that don't. But we are at the moment submitting that manuscript, and we also need an independent cohort to validate that paper. So there's still a lot of work to do. But I mean, it would be absolutely amazing if you could, when you get pregnant, come to your first visit at the maternal healthcare unit, and then you take a microbiome sample and you get to know if you have a higher risk or not for preterm birth, because then it could be kind of taken special care. You could be special recommendations on how to prevent preterm birth. Another study, just to mention, we also work a little bit with startup companies on the development of treatments for bacterial vaginosis. And I have one student, Emilia Lahtinen, who is an industrial PhD. Funded from SSF, and they develop antibiotic-free compound, which is called PIF, to treat bacterial vaginosis and fungal infections. And Emilia and we are looking into the clinical studies on the composition of the vaginal microbiome, and we see very positive results that this kind of antibiotic-free treatment that destroys the biofilm of the pathogens and lowers the pH value, that it really suppresses the bacterial vaginosis pathogens like Atopium vaginae or Fanny-Hessa, BVAB2 and Gardnerella vaginalis, and that Lactobacillus iners and Lactobacillus, the sum of all Lactobacillus species, really increases after a 7-day treatment. I didn't put it on the slide, but this is based on a study with 150 women with bacterial vaginosis. So to summarize, take-home message here, and this is a figure from a recent review we published, is that the microbiome may be a testable and modifiable factor in improving reproductive and long-term health for women. Lactobacillus crispatus is the species that is related to better health than other bacteria in the vaginal microbiome, but we also need large-scale cohorts with different ethnicities and with combination of standardized pipelines for sampling and analysis and lots of metadata around it to define the role of the microbiome in women's health. And it's actually not enough just to study the vagina in isolation. We need to study the interplay between both microbes and host, but also the interplay between the different parts of the body and the microbiome in the gut and the oral cavity as well. The vaginal and fecal microbiomes, together with individual risk factors, have the potential to be used to select women at risk to deliver preterm. And there are also new treatments needed to cure dysbiosis and to restore the microbiome, both like just based on disrupting biofilms and killing off pathogens, but also on restoring by adding the right bacteria to the vagina. And yeah, so with that, I want to finish and bring up my book here, which is also on the table. I think the female body is still understudied. We know so little compared to the male body, and there are diseases which are annoyingly less studied. For instance, endometriosis, which you probably have heard about, an inflammatory disease connected to menstruations, is as common as diabetes. 1 out of 10 has that disease, but we are decades behind in understanding its etiology and being able to treat it. So there is so much more research needed to understand the female body and the specific diseases of the female body. And there's also so much need for new innovation and access to better healthcare. And I think the first step is to start to normalize our bodies and break the taboos that are connected with them. Because one of the reasons why endometriosis is not so well studied is that probably young girls are just told that it is okay to have pain during menses and that you have to endure that. So if we start talking about it, if we kind of get also a very clear picture how to take care on our microbiomes and our female bodies, then we are kind of improving the situation already. And yeah, so this is the team. And we work at the Center for Translational Microbiome Research with Lars Engstrand heading it. And yeah, and that's with that, I thank you for your attention.
Thank you. And we're ready to turn off the recording. And can you have an eye out for questions from the online participants?